We are about to start. Sir, mute for the sound coming.
আগে গিয়ে পড়া ও আপনাদের স্পিকারটা কম। Good afternoon. Am I audible to all of you? Somebody please respond. Is it audible to all of you?
Yes, it is audible.
Okay. We are just about just give us 5 minutes. We are about to start. Our speaker is just about to join. So just give us few minutes. Be there with us. And also I'm seeing that 2 participants have joined with different names. One is 300125, another one is 3D Electronics. I would request those participants please change their name, and if you are not being able to change your name, let me know what your name will be in the chat box. I'll change your name by my end, because in this FDP this kind of names is not appropriate. So kindly let me know what is your name, or you change your name by yourself. 3D Electronics, kindly change your name or let me know what is your name.
Sir, my name is Jayanti Kishore Deb.
3D Electronics, 3D Electronics, is it your account?
No.
Hello, am I audible?
Yes, sir.
Oh, okay.
আমি করে দিই।
Okay, now my microphone is working. Okay.
Yes, sir. Yes, sir. We can also hear you.
Okay, okay. Now we can start.
Yes, sir. And if possible, sir, if you can switch on your video, if possible, so we can see you. Huh.
The light.
Yes, sir. You are visible and audible as well. No, sir. You are not audible. You have to switch on your audio.
Now audible, na?
Yes, sir. Yes, sir.
Okay.
You are absolutely audible.
Okay.
So very good afternoon, everyone. Thank you so much for being with us. We had faced little bit inconvenience and technicalities, but everything is solved right now. We are presently live streaming in our YouTube channel, JISU-PHARMA YouTube, and also our eminent speaker is already with us. So I would like to take the privilege to welcome you all from the Department of Pharmaceutical Technology, JIS University, in the 3rd day of our FDP Faculty Development Program. We are getting a very good response from all of you. We are getting good feedbacks from all of you, and you are responding in the feedback links after every day. And hopefully we'll get the same kind of response in rest of the days of yours. So we have 2 days more after this day. And now I would like to request one of our professor, Professor Pintu Kumar De, to come here and introduce our speaker to all of us. Sir, please.
Thank you, Sakhar. Very good afternoon to all the participants. First of all, I would like to take this opportunity to wish all the participants, delegates, and our eminent speaker a very happy Durga Puja in advance to all of you. Thank you. Let me introduce our today's speaker, Dr. Tawan Kumar Giri. He did his graduation, post-graduation, and PhD in pharmacy from Jadavpur University. He is very known to all of us. He is having 20 years of teaching experience, and he was associated with many reputed institutes, pharmacy institutes. And presently, he is working as an associate professor in the Department of Pharmaceutical Technology, Jadavpur University. As you know, his diverse research interests include polymer modification, hydrogel-based drug delivery system, solubility enhancement of poorly water-soluble drug, fast dissolving drug delivery system, reverse iontophoretic extraction of biomolecules, and nanotechnology. He had supervised around 30 MPharm students. He has completed 2 research projects. He has published around 70 research articles in the peer-reviewed journals and having 16 books in his book chapters. He has authored 3 pharmacy books and also edited 2. He has served as a guest editor of current chemical biology journal, Bentham Science. 2 of his research articles published by Elsevier received the distinction of High Cited Article Award. In the year 2017, he has received the Best Teacher Award of Maulana Abul Kalam Azad University of Technology. He received prestigious recognition, and the name has been included in world's top 2% Indian scientists for 2019 and 2020. I think this is a very brief introduction about our Giri sir. So welcome, Dr. Giri, to this 3rd day FDP program. The stage is yours, sir. You can start.
Thank you, Pintuda. So now I am.
Welcome.
Yeah, your screen is visible.
So my slide is visible.
Yes.
Yes, make it full screen and you can go ahead.
Okay. So, good afternoon, everybody. The Department of Pharmaceutical Technology, JISU University, organized that FDP program, and particularly theme is post-pandemic advancement, drug research, and sustainable development. So already 2 days over, and this is the 3rd day, and 3rd day's first session. So myself chosen that topic, particularly biomedical applications of pH responsive hydrogel. That is I giving the lecture of this particular topic. So that is very interesting hydrogel, and that hydrogel systems having different kinds are there, but I particularly enlighten pH responsive hydrogel and their applications in biomedical, means drug delivery, tissue engineering. Particularly I have to enlighten and focused. So hydrogel, every means body telling that thing. And everyday life also, we have to use up that hydrogel system. Basically, that hydrogel prefix is there hydro. Hydro means what? Water. So we can tell hydrogel means aqueous gel. But in the true sense, that is somewhat different. How they different? Means in that means that term aqueous gel means they told that they already solid, but not like that. Hydrogel having that cross-linked three-dimensional structure, and they can absorb huge amount of water or biological, and that can be retained. Means they not dissolve or hydrated like that. Means after absorbing water also, that they can be retained their structure. So that is the hydrogel system. So coming to the later, that is, you see that hydrogel system every day as well as in different fields that can be applied. You see gummy bears, disposable nappy, sanitary napkins, water crystal. So every day, that type of hydrogel system we are using, and we know that things. That gummy bear particularly made up of what? That is the collagen. Disposable, that is nappy, made up of the sodium polyacrylate. Sanitary napkin, that is cellulose, esters, and some other ingredients they are used up and making up of the sanitary napkin. And water crystals. Acrylamide polymer we know, and polyacrylamide, that is systems are there and making up of the hydrogen. So all these things, they can absorb huge amount of the water, and that water retain into their system, means in their three-dimensional systems. And that hydrogel based system in the agricultural field, nutrients and any other materials that can be supplied through the hydrogel based carrier. Means here that hydrogel used up as a carrier material, nutrients or other things we have to incorporate them, and in the that can be used in the agriculture. In the transdermal drug delivery system, means transdermal patch, that is also the hydrogel based system used up and incorporating the drug, and we can deliver the drug to the patient. And oral drug delivery system. Matrix tablet, this is also we can make out of the hydrogel based matrix tablet. Capsule, microparticles, nanoparticles, those we are giving to the oral route. So that is also hydrogel based drug delivery system we can use. Contact lens, you know, we are using. That is also made up of the hydrogel system. Wound care, different types of wound care materials available also in the market, and so many research work also going on. So that is also the hydrogel based film or materials. And big era is the nowadays, that is the tissue engineering purposes. Tissue engineering, what is the tissue engineering actually? Or regenerative medicine. Tissue engineering, suppose we in our body having different organ systems are there. In organ systems means different tissues are there. Nerve tissue, cardiac tissue, liver tissue, cartilage tissue, bone tissue, skin tissue. So different types of tissues are there. So that tissues means sometimes damaged. So what happened? To recover that damaged tissue or curing of that, so we can use up the hydrogel based system as a carrier material to deliver the cell to regenerate the damaged tissue. What happened here? That hydrogel based system used up as a scaffold material, and inside this scaffold means that polymeric systems, and giving some internal environment through which means where the cells we can seed it up, and that cell can be grown up to this environment. And where the damage occurs, cartilage, skin or other places, we can implant it, that hydrogel scaffold materials containing different types of growth factors, nutrients, and the cell. So what happened ultimately? That cell can be grown up, and ultimately, suppose cartilage tissue damage takes place. So we can sedate the chondrocyte cell into the hydrogel system, means that scaffold material, and that scaffold materials we can implant it to the damaged cartilage. So there, the cartilage cell or the chondrocyte cells are grown up and ultimately fill up that damaged portion. And that materials, the hydrogel system we are using, that is the biodegradable in nature. So ultimately degraded from the body. So in this way, that tissue engineering nowadays tremendous work, and marketed products also available, and also for using of the natural polymers that we have, means research is going on for that tissue engineering purposes, means as a using up of the hydrogel materials as a scaffold, and sometimes what happens, injectable hydrogels we have to use up, means that hydrogel polymer and cells are seeded and making about the solution. But where the damage occur, that solution we have to injecting out, and there are some pH or the temperature changes taking place, and that solution is converting into the gel form or the hydrogel form. That is known as the specifically in situ hydrogel. So that is used up for the tissue engineering purposes. So next important parameter, everybody having basic question in mind, everybody's mind having that questions. Solution means polymeric solution, gels and the hydrogel. What are the differences are there? Because in all three cases we have to use up the polymers, and that using up of the polymer, we can using up of the some solution we have to make up, sometimes preparing the gel and sometimes preparing the hydrogel system. So first coming to the solution. The left figure is the representation of uncrosslinked polymers. It's very, very important. If they have good affinity with the water, their meeting produces a solution. It means a liquid composed of polymer chains, each one completely surrounded by water molecules. Means that can be hydrated, and that polymer chains can be separated, and ultimately polymer means coming to the solution. If we increase the polymer concentration, then viscosity of the polymer can be increased. Next is the gel and hydrogel. Very, very important and confusing terms are. Means a common interpretation in polymer science is the use of the terms gel and hydrogel synonymous. Technically, gels are semi-solid systems comprising small amounts of solids dispersed in relatively high amount of the liquid. Gels are semi-solid system, means that but possessing more solid-like than liquid-like characters. Sometimes hydrogels are also described as aqueous gel because of the prefix hydro. Although the term hydrogel implies material already solid in water. Hydrogel means aqueous gel, means they are gel means what? They already, when we are taking out the water, putting the polymer solution, they solid off and producing the gel. And hydrogel also telling that is the aqueous gel, means the already solid in water. But in true sense, no, hydrogels are a cross-linked network of hydrophilic polymer. They ability to absorb huge amount of the water while maintaining their 3D conformity or 3D structure. But in cases of the gel, they are not maintaining that like that way 3D conformity or 3D like structures.
Hello, sir.
Hello, just creating problem of my slide because the next slide is not coming. So 1 minute.
Maybe, sir, your network issue. Hello. Sir,
Hello.
You can do one thing, you can stop share once and you can start again if that is possible.
It's not audible.
Sir, now is it changing?
No, not changing.
Sir, is it not changing? Hello?
No, not changing.
You are not audible even.
So now am I audible?
Sir, we can't hear you, sir. Oh, okay. Just a moment. Hello?
Hello?
Yes, sir, you're audible.
Sakhar, Giri sir is audible.
Yeah. Here there was a network issue. Yes, you're audible, sir. Sir, is it not changing your slide?
Just one slide is changed, but next not changing. When this is creating some, I don't know. So,
Sir, you can do one thing. You can stop share. And is it in your PPT mode, or it is in the PDF mode, sir?
PPT mode.
PPT mode.
ছোট করে আরেকটু নিচ সাইডে করতে পারবো।
Okay, now visible that my slide.
No, sir. You have to share your screen. Sir, if you send us the PPT, we can share from our end.
Okay, 1 minute, just I am trying.
Okay, sir.
Now visible. Hello.
Yes, sir.
Yes, sir, it is visible.
It is visible.
Okay, sorry for that interruption because there
No,
is
sir,
no
that's all
problem.
right.
This is also I feeling guilty. Okay, no problem. That is next slide. That is I am continuing. That is the solutions, gels, and the hydrogels. Next, gels which are polymeric network.
Sir, is it possible, sir, to make it full screen? Like, yes, sir.
So gels which are polymeric network already swollen to equilibrium, and addition of fluids results only in dilution of the polymeric network in cases of the gels. Although some of the gels are rigid enough to maintain their structure under a small stress, after exceeding the yield value, gel fluidity is observed with loss of polymeric structure. Suppose gels are there, so we are applying pressure. So after certain pressure, they can maintain their yield value. But exceeding that pressure means that yield value, they convert it to the gel form. means they lost their polymeric structure. When solvent meet the hydrogel structure, the chains are not able to separate and leave, but the affinity with the liquid let it enter more and more, causing a swelling of the three-dimensional network due to inherent cross-linking for hydrogen structure. Conventional gels can also develop small level of cross-links as a result of gain in energy, because means during the preparation of the gelling system, so that gained some energy, and during the shear forces. So at that time, small level of pseudo crosslinks may take place, but these crosslink reversible because of the involvement of weak physical forces. The basic framework both means that gels and hydrogels in the polymeric network you see in the figure. So in the above, there is the polymeric structure. Left hand side is the aqueous gel and right hand side is the hydrogel. So in cases of the aqueous gel, what happens? You see there is a hollow circles are there. So that represents the virtual crosslinks are there. But in the right hand side, hydrogel system, that solid lines are present. So that solid line represents the cross-linking are there. So that cross-linking may be physical or maybe the chemicals. Physicals means by heating or cooling, we can means there is a hydrophobic interaction, hydrogen bond formation, or there is a ionic interaction. So that type of physical means we can prepare the hydrogen. And by chemical means cross-linking agent, we have to use grafting, IPN. So all these we can made up by the hydrogen systems. So these polymers produce systems that span a range of rigidities, beginning with a solution and increasing to mucilage, jelly, gel, and hydrogen. Thus, hydrogen sometimes referred to as more rigid gel. So, history of hydrogen system. So, in 1889, the term hydrogel was first described by Lee. For their terms giving that time the colloidal gel. Colloidal gels are formed by the aggregation of some micron colloidal particles, means positively charged and the negatively charged particles are there. They are self-assembly and making of the through the electrostatic interaction, they are forming the colloidal gel. And in 1914, first hydrogel for biomedical implant, that is polyvinyl alcohol, cross-linked with formaldehyde and marketed with the trade name means Evalon. So that is the first hydrogel for the biomedical application. And in the 1960, that is the big revolutions of the hydrogel system. That is poly-2-hydroxyethyl methacrylate, p-hemagels for the contact lens, that is discovered by with Charlie and Lee. That is revolution for present day hydrogen. That is cross-linked networks, swelling without dissolution, safe, they can be retained or mended. And 1970, this is also the revolutionary of that hydrogen system, that is stimuli sensitive hydrogen. And till that stimuli sensitive also used up nowadays. That is means suppose some hydrogels are there, that is unsolent. Means if we apply external stimulus, suppose temperature, that is temperature responsive hydrogen, ionic strength responsive hydrogen, pH responsive hydrogen, light radiations responsive hydrogen, ultrasonic radiation responsive hydrogen, magnetic field responsive hydrogen, electric field responsive hydrogen, urea responsive hydrogen, metal responsive hydrogen, antibody responsive hydrogen, glucose responsive hydrogen, morphine responsive, means these are responsive hydrogens, means external stimuli that have to be applied into the hydrogen system, and that can be used up as a carrier materials for delivery of the drug or for the delivery of the cell, and numerous applications they have. So next, it's coming to the what are the hydrogel definition. So hydrogel is cross-linked network structure that is swell in water and hold a large amount of water while maintaining the 3D structures. Means main thing is that is cross-linked polymeric chain, and they can absorb huge amount of the water or biological fluid, and after absorbing, they maintain their 3D structure. And water must be at least 10% of total volume. That may be 100%, 200%, or 300%, means times they can absorb water, but minimum 10% of the total volume of weight they must absorb. Means then we can tell that is the hydrogen system. And cross-link junction points where more than two polymeric chains cross over. Means cross-linking point means that is 2 or 3 polymeric chains they cross over, means each other. And we have told that hydrogen system absorbing huge amount of the water or the biological fluid and retain there. Why they absorb the huge amount of the water or the biological or the aqueous fluid? Because in every hydrogen system that contains the polymer, and that polymeric chain containing different hydrophilic functional group, NH2. NH2 where? Chitosan polymer is there. That polymeric chain containing the NH2. COOH means that is alginate. That is the gyrangam, sodium carboxymethyl cellulose. All these polymers having the COOH. OH having all polysaccharides, CONH2. That is, and the CONH, that is the acrylamide polymer. They are also be that type of functional group. SO3H, that is the carrageenan. So these type of functional groups are there in their polymeric structure of the hydrogen. That's why they are absorbing the huge amount of the water or the biological. Next is the very important question, resistance to dissolution. But that hydrogen system not dissolve. Why? Because of that cross-linking. So that cross-link that can giving up of the structure of the 3-dimensional structures like that. And that dissolution of that hydrogen system depend on the cross-linking density, means how much density of that cross-linking portions are there. And ultimately, that dictates the dissolution of the hydrogen system. Water can be free to diffuse or be loosely bound or tightly associated to the network. That is the cross-linking hydrogen. Previously, we have told. Other means hydrogen means that is the cross-linking. That cross-linking may be physical or the chemical. Just here giving two example means picture, nothing but in the literature, if you're getting about the so many physical cross-linking there and the chemical cross-linking. The right hand side, you see the physical cross-linking means that is just forming the secondary hydrogen bond. So that there may be the hydrophobic interactions also, or ionic positive and negative interactions also. That is means all these are the physical cross-linking. So you see two polymeric chains are there, and between these two polymeric polar groups, there is a hydrogen bonding are there. Means there is a physical cross-linking takes place. And another is the chemical hydrogens. Chemical hydrogen means chemical cross-linking to the covalent bond between two polymeric chains. Means here we have to use the cross-linking agent. Cross-linking agents means that linking of between two polymerics or the 3 polymeric chains like that. And they're making out the covalent band. And you see the right hand figure. So there are 2 polymeric chains are there. And that 2 polymeric chains are covalent cross-linked. So that is the, and forming the hydrogen system. So that is the chemical hydrogen systems. Next is the advantages of the hydrogen system. So first advantages of the hydrogen system, structural and compositional similarity to extracellular matter. Extracellular matter means in our body or the biological system. So that extracellular matter is soft and tissues are there. So that hydrogen system when absorbing the water, that is just like a tissue-like structure. Means there is a similarity is there in the extracellular matrices. And extensive framework for cell proliferation and survival, for particularly regenerative medicine purposes or the tissue engineering purposes. So there what happened? We can be that hydrogen system used up as a scaffold material, means that is a carrier material, where we can keeping up of the nutrients, growth factors, and cell we can see data. and the cell can be grown off into this medium. So that hydrogen system is best carrier system or the scaffold for cell proliferation and the survival, because they giving about the watery structure and the nutrients, growth factor, all can be stayed along with the that type of cell that can be grown up. Next is the non-thrombogenic. Suppose some materials, if we enter in our body, so sometimes what happens? Blood clotting taking place. This is giving up what? Thrombogenic action, because external materials will enter into our body. So they sometimes giving out the blood clotting like that. But in that case is the hydrogen system. So they are giving up of the non-thrombogenic properties like that. Next is the biocompatible. One word, very, very simple word, but very big word is the biocompatible. Means in our biological system that is compatible or not like that way. Means biological system means that is different from the other systems like that way. Suppose your biological system and my biological system, totally different system. They are working in their own ways like that way. So that type of systems or the matrix or the carrier, whatever may be, that can be compatible in our biological systems. So they can tell that the hydrogen systems are biocompatible. Means maximum most of the cases that polymer we have to use that is biodegradable, means biocompatible. Next is the biodegradable. Suppose some materials we can be incorporated into the implanted in our body. So if they are not biodegradable, so how they can be removed from our body? Means that polymeric structure ultimately converting into the body into the monomeric form, carbon dioxide, water, or that is that type of small molecules like that. And that molecules can be easily removed from our body like that. But that hydrogen system, that polymeric material used that is the biodegradable, means degraded inside the body and ultimately removed from our body. Next is the good transport of nutrients to cells and products from cells, because cells tissue engineering capacity. Suppose the cells are there and that environment nutrients or the other materials or that is means some materials are required. So that materials can be transported to the cell, and cells producing suppose toxic materials or the byproducts like that. So that can be also transport through this hydrogen system and can be injected in vivo as liquid that gels at body temperature. This is very, very important portions of the hydrogen-based drug delivery system. Injected means that is in situ gel forming drug delivery system. Means we have to inject it at the hydrogen means polymeric solution form. And after insertion into the body, where requirement is there, they convert it into the gel form of the hydrogen form. So that is means easily we can administer to the body, because they maintain their solution form. But after entering into the body, they are converting into the gel or the hydrogen form. And hydrogen system also mucoadhesive and bioadhesive properties are, because you know so many polymers we are using up here. So that polymer itself having the mucoadhesive characteristics are there. Means mucins are there, they are interacting with that polymer, directly interacting with the mucin components and ultimately adhere there. Means they giving up or show off of the mucoadhesive characteristics. Next, coming to the types of hydrogen. So in the base of method of preparation, the hydrogen, homopolymer, copolymer, and interpenetrating polymer network. You see the topmost left hand side figure, homopolymer means same polymeric chain. Suppose acrylamide is there. Acrylamide, they are cross-linking each other and forming the polyacrylamide. Means one monomer, they are cross-linking like that way and getting above the. This is the homopolymer. Copolymer means 2 or 3 monomers, they are cross-linking with each other and forming up of the that is the homopolymeric copolymeric systems like that. And that copolymeric system is the that is the alternate copolymer, then random copolymers like that. And that is means the polymeric chains or the monomers, how they arrange like that. And in this way, they are forming the means copolymer hydrogen system. Next one is the interpenetrating polymer network. This is very, very important for those who are being in the hydrogen based system. Then IPA, interpenetrating polymer network. But there are two terms. One is the semi-IPN and another is the IPN hydrogen. I giving simply example of you to understand what is the difference between the semi-IPN and the IPN. Suppose you take the alginate and another polymer, chitosan. So you make up the solution of the alginate and chitosans like that and put into the calcium chloride solution. Then what happens? Calcium that is interacting with the sodium alginate containing COOH group. So crosslink thing taking place. So they forming the calcium alginate. But chitosan is not crosslink, but they are forming the hydrogen. That is known as the semi-interpenetrating polymer network. Two polymeric networks are there. One is the sodium alginate and another is the chitosan. But here the alginate is cross-linked with the calcium, but chitosan simply present as a polymeric chain. So that is the semi-interpenetrating polymer network hydrogen. But in cases of the IPN or the full IPN, suppose we are taking off of the alginate and gelatinum, both containing what? Natural polymer, polysaccharide, COOH group. So we make the solutions of the sodium alginate and the gelatinum and put into the calcium chloride solution. Then what happens? So gelatinum containing COOH, they cross-link with the calcium. Alginate containing the COOH, they cross-link with the calcium. Then what happened? Alginate and the gelatinum independently cross-link and interpenetrated. So that's why that structure is known as the full IPN structure. So next is the based on environmental responses. So previously I have told there is so many environmental responses are there, and that can be according to that environmental, they can be swollen up and giving the responses. pH, light, temperature, electric field, so many glucose are there. So they are according to their responsiveness, they can be classified. Next is the pH sensitive hydrogen. This is my particularly 2 days lecture. So that is the pH sensitive, because so many hydrogen systems are there. So that is I unable to discuss because 1 year required to discuss all the hydrogen systems like that. But I giving up some outline or the basic of that hydrogen system. But the pinpoint is the pH sensitive hydrogen system. pH sensitive hydrogen system is what? The hydrogen system which responses to the external stimuli, that is the environmental. That hydrogen is known as pH sensitive hydrogen. Means if the hydrogen system respond particularly the change of pH, that type of hydrogen system is known as the pH responsive hydrogen. Means systems that can respond to a dynamic pH environment are of particular interest for biomedical application. At several locations in body, it is substantial pH change during either normal function or as a part of the system, because in our different organ of the different parts are there. So different organ and the different parts are in the different pH systems are there. So accordingly, we can modulate or develop that type of hydrogen-based system to deliver the drug or for the tissue engineering purposes. Means there is a lot of exploration part is there for the development of the pH responsive hydrogen. Blood, stomach, upper small intestine, colon, tumor, early endosome, late endosome, vagina, inflamed tissue. They are having the different pHs are there. You see the tables are there. Means so accordingly we can target the or deliver the drug through this hydrogen based system to that of the particular organ. Next is the polymer composition and pH responsive swelling layer. How they are swelling means that how much polymer are responsive means because all the polymeric means those are ionic characteristics, suppose pendant functional groups are there, that is cationic or anionic. So accordingly, means when exposed to an aqueous solution of appropriate pH, ionic state, these pendant groups will ionize and result in the buildup of fixed charge along the polymer. The generation of electrostatic repulsive force results in the pH dependent swelling and deswelling process as the water is either absorbed or expelled from the hydrogen. You see in the figure that is in the x-axis pH and equilibrium swelling. So two types of curves here. First, suppose giving about the anionic hydrogen. If increasing the pH certain level, then there is no change of swelling status. But after that, the swelling is increased. Means up to the pKa value of that anionic groups of the hydrogen system. Means above this pKa value, that ionic means hydrogen system containing the ionic group that can be ionized, and that ions repulsive, giving off of the means repulsive force they create and ultimately swell up. Giving suppose anionic hydrogen made up of the alginate, sodium alginate. So containing what? COOH group is there in the stoma, or they means not ionized. But when reaches into the small intestine, so in the basic pH, that COOH group means ionized and producing the COO minus, and they can be repulsed each other and ultimately swell up. So you see, after certain point, they can be the swelling status of the polymer system can be increased. And another cation polymer, that is in the low pH value, they giving about the highest swelling characteristic. Suppose the chitosans are there, amino group is there. So low pH what? Protonated means NH4 plus they are creating. So they are repulsive each other, and ultimately the hydrogen system can be swelled up. But after certain pH value, they can be means deprotonated, and that is not ionized, and they can be deswelling of the hydrogen can be takes place. So the anionic hydrogen, you see, when pH greater than that is the pKa value. pK value when upward in the acidic solution, you see just shrinkage, sodium alginate. And in the basic medium, you see OH minus are there. Ionization of the anionic hydrogen taking place, alginate, and that can be swollen. And below figure, you see the cationic hydrogen. In the acidic solution, what happened? Protonation, H plus is there. Ionization of the the basic groups taking place and swell up. But in cases of the basic pH less than, that is the not swell up, but particularly the chitosan hydrogen system. So that pH responsive hydrogen here giving off of the here chitosan, guar gum, alginic acid, hyaluronic acid, carboxymethyl dextran, carboxymethyl cellulose, gelatin A and B, tertiary amine starch ether. So all these are chitosan NH2 functional particularly. And this is swell up in the acidic pH. Guar, that is guar gum, COOH, basic pH. Alginic acid, COOH groups are there. In the basic pH, they are swelled up. Hyaluronic acid, COOH groups are there. In the basic environment, that can be swelled up. Carboxymethyl dextran. But in cases of the UC, gelatin A and B, here both the groups are their pendant group. One is the basic group and the acidic groups also there. Means NH2 and COOH group. So below pH means NH2 group of pK. So that can be protonated and ionized, swell up. And when reach the basic pH level, COOH groups also there in the cases of protein. So in that cases of the swell up. So that protein particularly swelled up in acidic pH as well as at particular basic pH, but depending upon the pKa value of NH2 group and the COOH group of the gelatin. So next is the suppose giving some pH responsive polymer, that is you see the chitosan, N-acetyl glucosamine and the glucosamine. NH2 groups are there. So only at acidic pH, that is deprotonated, and neutral pH they deprotonated, but acidic pH they protonate, and pK that is the 6.5. So how we can prepare the hydrogen system using of the chitosan? Simply you have to chitosan dissolve into the acidic solution and making of the solution, and another solution you have to take up the counter ion. So that is the tripolyphosphate, and ion you have to. And pour there, that is the ionotropic gelation takes place, and they are that interactions, and they're making of the hydrogen system. And you see that hydrogen system, and that swell in the low pH value, pH 1.2 means protonated, and from there the drug can be released. But in basic pH, pH 7.4, that is unswollen condition, and drug not released from the systems like that. And another you see the guar gum, that is the non-ionic polysaccharide, means they are not giving up of the pH sensitivity. But that particularly guar gum we have to modify or the derivatives. If we introduce in the acidic group, so what ultimately coming to the pH sensitive natures like that. So some researcher, they developed guar-gum succinate like that, means acidic group they enter, and ultimately develop the delivery system for colon targeted drug delivery system. Means at higher swelling, greater drug release were observed, means in the SIF, then in the SGF, simulated gastric. And next, carrageenan also one of the polymer. So they are kappa carrageenan, iota or lambda carrageenan are there. So they are different in their sulfate group. In the kappa, 1 sulfate group, and iota, that is 2, and the lambda, that is the 3 sulfate groups are there. In the they differentiate, and that this is the anionic polysaccharide, and they can be made up of or used up for the pH sensitive hydrogel based system. One of the researchers, they prepare chitosan carrageenan hydrogel base, and that cross-linked with the glutaraldehyde, and ultimately developed for the drug diclofenac sodium, they can be incorporated, and the drug release extended up to 24 hours, and the maximum drugs that can be released into the intestinal pH, in the basic pH like that. Dextran also, there is the natural polysaccharides, and that is dextran particularly for the colon targeting or the colon specific drug delivery system, that polymer can be used because in our colon, so some enzymes are there. So that enzymes degraded some polymer. So dextran is one of the enzymes present in the colon. So that particularly degrade the colon and degrade the dextran. So here different types of cross-linking agent and another polymer also used up for the making of the hydrogel system. And for that hydrogel based system enter the BSA, bovine serum albumin, and ultimately that can be deliver the drug to the intestinal region. Next is the xanthan gum. Xanthan gum is the ionic polymer, and that is the carboxylic groups are there. pK value of the carboxylic group, particularly 4.6, and that is also anionic polysaccharide, and that can be made up of that is the hydrogel pH sensitive hydrogel system, and used for the drug delivery process. So next is the alginate, because we are not discussing too much about that of the polymer, but here also alginate is one of the anionic polymer used for the development of the hydrogel based, means pH sensitive hydrogel based drug delivery system. So here we can giving up some examples of that hydrogel based drug delivery system, how they prepare and develop. For the intestinal drug delivery system, one scientist or researcher prepare the pH sensitive graft copolymer of the polyacrylamide and sodium alginate, and ultimately develop the hydrogel based system. Here sodium alginate is taking up, and ammonium persulfate are used up as an initiator. Grafting means one main chain of the polymer is there, and to there we are attaching some another monomer, and in this way big branched chain polymer we have to prepare. There are certain advantages also there. Suppose in cases of the natural polymer, so they having some drawbacks are there, means they are easily hydrated or swell up, and releasing the drug in a faster manner, or microbial contamination may take place. So in this way, we have to graft it or amalgamate it with the other polymers to that of the natural polymers like that. So here also for the drug delivery system developed purposes, so they can be grafted, means ammonium persulfate we have to use up the initiator, and they initially create the macroradicals, means OH site of the polymeric chains, here sodium alginate, then acrylamide monomer we have to add on, and to that of the alkoxy macroradical site, that acrylamide chain attached there. But after that we have to hydrolyze that polymer, because that CONH2 group here, so that making the anionic character we have to hydrolyze with the NaOH, and ultimately that CONH2 group converting to the COONA, because here that scientists prepared particularly the hydrogen system cross-linked with the calcium chloride and making up of the anionic hydrogen to deliver the drug to the intestinal site like that. So that's why after grafting that can be hydrolyzed. And that hydrolyzed polymer we can taking off or dissolve into the water and putting into the calcium chloride, and ultimately through ionotropic chelation method, and they can be make up the pH sensitive hydrogen peak. And that developed hydrogen peaks show up the pH sensitive nature. So initially you have to see in the pH 7.4 that soil here. Then that same hydrogen system putting into the pH 1.2. So de-soiling techniques. Then again put into the 7.4 soiling. Again pH 1.2. So this is means sort of the pH sensitive nature of that developed hydrogen system. And that drug delivery system, they can be giving up the in vitro releases, they can be swapped, and you see the figure there in the below pH 1.2, only minimum quantity of the drug reach up to 4 hours, 10%. But in cases of the pH 7.4, in the intestinal pH, the 100% drug release within 8 hours like that. Next is the another pH sensitive hydrogen based system for colon specific drug delivery system. Here what happened, that acrylic acid based hydrogen system we have to manage both side acrylic acid and cross link with the azo polymer, means azo compounds that can be cross linking. Azo means two nitrogen compounds are side by side are there. So that acrylic acid polymer cross linking with that of the azo compounds like that. So when pH means in the stomach, that hydrogen system, so low soiling, no drug release taking place. But in case of the intestine or the small intestine, pH increases, so there is the ionization of the COO2 takes place. But the drug is not released because that is the azo groups are there. So that is cross-linked because that azo groups only hydrolyzed in the colonic enzyme, that is azo reductase are there in the colon. So that degrade the azo bonds, and after that the hydrogen, the polymeric systems are separated, and from there the drug is releases into the colon. And another is the stomach specific, suppose some drug delivery, some requirements are there in the stomach cancer or the Helicobacter pylori. So that is reside in the stomach. So our sometimes the requirement of the motto is there to deliver or target the drug specifically into the stomach. So in that case, if we develop the pH sensitive drug delivery system, so we can target there. So here here one thing, not stomach, we can deliver the drug to the intestine. Next slide, I coming to the stomach, because insulin is the proteins that is degraded or decomposes by the host environment of the stomach. So that host environment of the stomach, that we have to bypass. That's why we have to made up the pH sensitive hydrogen based system and incorporating the intestine into there. And when the hydrogen system pass through the stomach, so that insulin not released and protected there. And when reaching into the intestine, so that can be swollen and the insulin can be released from there. So in this way, the insulin can be degradation of the insulin can be protected. So in the host environment of the stomach. Next is the tumor targeted, suppose drug delivery purpose. Suppose we have to target the drugs to the tumor site, and particularly tumor targeting, there is a active targeting is there and the passive targeting is there. Passive targeting means normally develop the drug delivery system and in the blood, and passively different types of that is the phagocytic systems are there, MPS system or the macrophage system, so they can be taken up naturally. That is the passive targeting. And another is the active targeting. Active targeting, where the cancer is there, we can target the drug, means anti-cancer drug to there. Means suppose in colon cancer is there. So here what happened? CD44 receptor is overexpressed, or any other portion of the cancer is there. So some receptor is overexpressed. So we have to attach the ligand material to that of the carrier material. Means they can be carried to that of the particular positions. So where reach to that tumor positions and tumor microenvironment also having some pH variation. In the extracellular position, slightly acidic in nature. but inside the cancerous cells having the slightly basic in nature. So if we develop some pH sensitive hydrogel based system, so if we that swell up into the acidic environment like that way, so there the drug may be released like that in the extracellular matrix, and after that the drug can be enter intracellular and kill off like that way. So cationic hydrogel ultimately target to the extracellular portion of the cancerous cell because slightly acidic in nature. But if you target to the drug intracellular, so the drug should be incorporated into the anionic hydrogel system. But so that particular hydrogen system only releasing the drug, but not they reaching, means they are not carrying. So for carrying purpose, carrier purpose, means we have to incorporate some ligand material, means where that type of receptors overexpress. So that type of receptors attached to that of the pH sensitive hydrogel system, they can carry and reach the hydrogen to that of the extracellular portion, and where the slight pH changes are there, and that can be swell up and releases the drugs. So here giving one example. So both natural and synthetic polymers we can use up. One scientist, they using chitosan derivative to hydroxy 3 ethyl ammonium propyl chitosan chloride, that ionically crosslink sodium triphosphate simply and making of the cationic chitosan based hydrogel system, and that can be incorporated the drug methotrexate and ultimately deliver, and in the tumor side the drug is released. And next is the tissue engineering purposes. You see that right hand side of the figure. So suppose cartilage tissue or bone tissue is fractured. So from there we can taking off the cells ultimately separated. Then we have to cell proliferate the cell, and that cell we can incorporate into the hydrogel system, and that hydrogel means injectable hydrogel and injected into the fracture portion. What happens after entering the hydrogel in solution form? They are gel, they are forming 3-dimensional structure, and inside this having what cells are there grown up, and ultimately the polymeric hydrogel system biodegradable. So in this way the tissue can be repaired. This is big era of the that hydrogel based systems for the regenerative medicines, and that is a lot of research going on particularly for that. And one giving just example, pH and thermo, both sensitive hydrogel here used up one scientist, and for tissue engineering purposes they can be used up. So that sulfamethaxine poly-ecaprolactone collectide polyethylene glycol poly-ecaprolactone collectide, this is copolymer. They developed that polymer exhibited both temperature and pH responsive behaviors like that. So here the important criteria is that in temperature 10 degrees centigrade, pH 7.4, that is solution. pH 8 also solution, but you see the temperature 37 and pH 8, that is in the solution form. So when that type of hydrogel system, if we injecting into the body, if we use pH 8 and the temperature 37 degrees centigrade, then what happened in the solution form? So after injecting into the body, reaching there, so temperature 37, pH 7.4, so they are converting into the gel form like that. So that researchers are performing some in vivo study, means that solution injected subcutaneous, and that injection or the solution form containing human mesenchymal stem cell and RH bone morphogenic proteins like that. So to grow up of that cell, mesenchymal stem cell, RH BMP used. So you see after 7 weeks that hydrogel structure in the back side of the mines, they can be retained there like that. So what happened? So if inside having some cells are there, so that cell can be grown up like that, and after that the time being the polymer can be degraded and removed from the body. So next my final point, that is the conclusion, that is research in the area, particularly pH sensitive hydrogel, both the drug delivery and the tissue engineering field well established, and the pH sensitive PUBR can be engineered by grafting or co-polarizing certain anionic or acidic monomers or cationic groups such as quaternary ammonium groups on polymer chains, and can be used for site-specific drug delivery and tissue. So this part is exploration site. You can explore tremendous exploration sites that are engineered by grafting or co-polarizing. So you can develop new polymeric hydrogen system, because in our nature, having so many polymeric systems are there. You can extract also, and you can modify and develop to the new engineered polymeric materials. So you can, so this is the big exploration sites for our research. Many hydrogel-based drug delivery and tissue engineering devices have been designed, studied, and in some cases even patented. However, not many have reached the market. Some marketed products also available, but so many marketed products are not also available based in the hydrogel system. As research continues for the development of hydrogel-based drug delivery system, we'll definitely see significant advances in the coming decades. You can develop in both healthcare and industry that arise directly from hydrogel and tangibly improves the world. References and some of my particular pH responsive hydrogel based system, I have developed some work and published in the some paper, and this is my two hydrogel related book already published. One is CRC Press and Elsevier, and now also one hydrogel based book, also editing work is going on. It's probably published in the 2023. Thank you, and particularly thank you for the my classmate, particularly Dr. Himanshu Shekhar Maji, that is the head and the academic director of the GIS University, and the organizing committee of the conferences, Dr. Amit Arindam Maiti, Dr. Ritu Khara, and Dr. Abdul Amin. Sorry, going to be within 1 month, maybe the doctor. So once again, thank you all for your patience, because some problems happened during my presentation. Okay.
That's absolutely fine, sir. And thank you so very much for delivering your knowledge and your research with us. It was, I'm sure it was because in our university, in our department, a lot of researchers, scholars are doing their research on hydrogel. They're taking hydrogel as a delivery system. And I'm sure they have got benefited by that. And now I would like to request the participants, if you have questions, you can even unmute yourself and ask the question. If it is not possible for you, you can write the question in the chat box. We can take the question from there. And I'm sure the sir will be very happy to answer that. So the platform is open for question and answer right now. And those who are watching us from our YouTube channel, JISU-PHARMA, so they are also they can also ask their question in that YouTube. We can take the question from YouTube as well. Sir, right now we have two questions from our end to you. I would like to say that to you.
Okay.
Sir, I would like to know that what are the most basic characterization that needs to be done for the hydrogel?
Okay. So, particularly for the hydrogel based system, here giving one basic thing. Several times I have told hydrogel is the cross-linked structure. So that cross-linked structure means after manufacturing or the preparation. So that hydrogel system we have to first characterize through FTIR or the NMR study. Means there is any cross-linking takes place or not, like that. Because if there is no cross-linking taking place, so how we can tell that the system is the hydrogel system? Suppose here I am giving another important point. Some materials itself having crosslink structure are there. So if we develop the solid dosage form like that tablet or any other, so their materials itself utilize and they retain their structure and they come in when come in contact with the whatever the biological system, they can be swayed. But if we making of the solution of that crosslink structure by applying heat or force or any other, so that may be degraded that crosslink structure. So that is the question. Means after preparation, we have to first characterize through means the FTIR or the NMR or any other XRD or DSC studies are there. So there may be any linkages there or not like that. So this is the one. Second one is the for the swelling studies also there. Means how much capacity of the fluid or the biological fluids or the water. So that can be absorbed like that. So if we, for the drug delivery purposes, we have to deliver the drug like that. So drug entrapment efficiency we can determine. Means how much quantity of the drug that can be entered. And the drug release study, so that can be, we have to determine like that. So in this way, or any other so many characterization means similar to that of the general drug delivery system, we have to evaluate or characterize that.
Right, sir. Sir, there is few other questions in the chat box. So Shayan Bishash is asking, what is the long-term toxicity potential of these hydrogels?
Long-term potential study of this hydrogen-based system, because this is the very important question for the toxicity point of view, because we have to use up the different polymeric systems here. If we used up for the natural polymer system, natural polymer or the polysaccharide systems itself. So that is, we know we are in the food materials or regularly we have to take up this material. So they are not showing up of any toxicity or like that. And for preparation of the hydrogen system, if we used up for the any chemical crosslinkers like that, so there may be some toxicity concerns are there. But if we making of the hydrogen system by physical means, so there is no need to use up of the chemical crosslinker. So that is absolutely free of that toxicity like that. But in another, suppose if we used up some new polymeric system, synthetic polymers or like that, so there may be the question of toxicity. And when we have to develop this system, we have to study the toxicity, acute and chronic, to characterize that new polymeric systems toxicity like that.
Okay, sir. Sir, another question is there from Dr. Kiran Kumar. He's asking, what is the extent of mucoadhesion by hydrogels?
Extent of mucoadhesion means how long they can be adhere to this our mucus system like that. So that is actually means which type of polymers we have to use now and which type of hydrogen based system we have to develop depending on their means nature of the polymeric system that also can defend, and also their cross-linking density. So that cross-linking density also determine how much they have to swell down and how much they have to adhere to the mucus membrane, and that mucin containing which group, means that some groups are there. So if we opposite to that group of the groups we are incorporating into the hydrogen system, so in that case the attachment of that hydrogen to that of the particular mucus should be extended or for long time like that.
Okay, sir. Sir, another question we have from our end. For polymeric reaction, why nitrogen atmosphere is very much important, and that we want to know, sir.
Particularly, this is actually this is the means synthesis part, means chemistry part actually, means we are using of suppose grafting or synthesizing polymer in our laboratory. In particularly conventional method, we have to use the nitrogen, means to create the inert atmosphere into the system. So otherwise, if oxygen is present into that system, so they may react with the because when we particularly grafting we use the polymer, we have to use the monomeric system and we have to use the in the initiator system. So that react with each other, means in the presence of oxygen, and they may be producing the toxic products like that. So that's why we are creating the inert atmosphere in the during the synthesis of the polymer or the grafting of the polymer.
Thus, oxygen won't be able to react. Okay, sir. Sir, I hope there are no more questions, and I think the way you have explained things to us and few questions we had from our end, as we are also in our department trying to work with hydrogels. So I think this kind of information would have been very helpful for us as well. So, sir, with the permission of you and with the permission of the coordinators of this FDP, our department has a very small token of appreciation for you, though this token is very minimum for in respect of the way you have explained things to us. You have given your time, and with your permission, I would like to show that on my screen.
Okay.
So we are really thankful for your cooperation in this FDP, sir, and being a part of this FDP we are really very much privileged to have you amongst us.
Okay.
And we'll be sending this to you through in hard copy by post, sir, as well.
Okay. So thank you once again, all of you. So can I leave now?
Sure, sir. Sure.
Okay.
Absolutely.
Okay.
Absolutely. Now I would like to request the participants that I would like to mention that we are about to start the next session of ours. Our next speaker is also with us. I would like to request one of our faculty members, the Assistant Professor, Dr. Payal Chakrabarti, to come here and introduce us with the second speaker of today. Payal, ma'am. Just a moment. I have my shoe.
A very good afternoon to all. So we are here for the second session of today's FDP. So firstly, I want to introduce our next speaker for the session, Mr. Kumar Lahiri. He was actually a very dynamic and pharmaceutical professional with a 16+ of experience in the industry, and specifically in the domain of formulation development and product technology transfer. He had 5.5 years, almost you can say 6 years of experience in generic formulation development from solid dosage forms, sterile and novel drug delivery systems. Almost 10 years of experience he gathered from technology transfer, process development, and different validation of drug products of solid oral for highly regulatory markets like US FDA, EU, WHO, etc. Previously, he was serving from one of the most eminent pharmaceutical industries, the Mylan Laboratories, where he served as the Deputy General Manager in Technical Services Department in the Indore plant. And presently, he is working with Dr. Reddy's Laboratories in Hyderabad as a lead process development. He handled successful technology transfer of more than 70 drug products, well versed with formulation development, process optimization, process validation, post-validation, technical troubleshootings, and site transfer and alternate vendor qualifications. He had been very proficient in managing his day-to-day operations for process evaluation of newer products, validations of launch batches, handling of incidences, deviations, manufacturing investigation with adherence to the quality systems. Successfully, he has been faced various international quality audits, such as US FDA, MHRA, WHO, and PMDA from Japan. His most areas of expertise lies with the process development, scale-up, technology transfer of the product. He had hands-on expertise in providing technical inputs during product acquisitions, product transfer, and other techno-communal activities, commercial activities. He had been expertise in evaluation of various novel technologies like booster process for pellet manufacturing, hot melt extrusion, and solid dispersion technology for enhanced bioavailability of the products, and dry and wet granulations, etc. He had very sound experience in handling of multi-unit particular systems and extrusion spheronizations technology. That's all in a short CV of Sir. He had definitely gained a lot of experience from the industry because it's a vast time where he has spent most of his period working with the industries. So next, I would like to call upon on the dais our next speaker, Mr. Ashit Kumar Lahiri. He can be with his presentation.
Yeah, thank you, Payal, madam, for your nice introduction. Okay, so probably you can see me. Am I out? Hello.
Yes, sir, you are audible.
Okay, so thank you for a nice introduction. Okay, and thank you for JIS team for giving me this opportunity to share my experience with the team. Okay, and so I can start now, or?
Yes, sir, you can start your presentations.
Yeah. Is it visible, my screen?
Yes, sir, it is completely visible.
Okay, so good afternoon all, and today actually I'm going to give a presentation on this application of PAD tools and digital process in pharmaceutical research and manufacturing. Okay. So first I will explain about the PAD tool application in pharmaceutical manufacturing. What is PAD and how we are applying this technology in our day-to-day pharmaceutical manufacturing. Although this PAD tool application, it is a very recent technology, and many companies, means advanced, many advanced companies, pharmaceutical companies, they are only utilizing this PAD tool. Okay. So first I will explain about the PAD. What is PAD? So PAD is an important part of much broader and risk-based paradigm introduced by 21st century current good manufacturing practices. So FDA has adopted a paradigm shift in its thinking in current expectation. So what is the current thinking of FDA? They have actually switched from our this empirical and univariant experimental development to QBD based development, and expectation is for with respect to validation, that is conventional validation is shifted to continuous process verification. Similarly, these process controls are expected to shift from offline analysis at QC to real-time testing during the process manufacturing of the batch. That is the expectation. So in order to do this online testing or real-time analysis instead of doing going for the offline testing in the QC, so this the need of PAD tool came into the picture. How to apply PAD? New technologies. So how to apply PAT, new technologies in pharmaceutical manufacturing. So PAT has been defined as a system for designing, analyzing, and controlling manufacturing through timely measurement of critical quality and performance attributes of raw and in-process material and processes with the goal of ensuring final product quality. So this is the definition given by FDA. So this is a designing and analyzing and controlling of manufacturing through timely measurement. Timely measurement means, like for example, we manufacture some any product, start from the dispensing to the finished product. So in between, there are different process stages up there. So so using this PAT tool, we can test our in process control, and then performance attributes, including raw material and in-process material and processes. So that is the goal. What is the principle of PAT? PAT principle is quality cannot be tested into the product. It should be built in or should be by design of the product. So what is the goal of application of PAT? So the main goal is to identify the defects in process manufacturing, reduce the process variation, enhance the process safety, and then produce the product quality information in high time, building quality into products and enhance understanding and quality of understanding and control of manufacturing process. So what is identity? What is identify the defect in manufacturing process? So in manufacturing process, there is some in-process control, like measurement of the temperature, measurement of the measurement of different coating parameters. So if we can control those parameters, so then we can ensure our finished product to meet our target quality performance. Okay, so this is the main objective. Identify the defects in the process manufacturing, then reduce process variation. So in order to reduce the variation in our process, we are applying this PAT tool in the pharmaceutical manufacturing. Then enhance the process safety. Enhance the process safety means there is many process safety measures are there which can be ensured through this PAT tool. Then produce the product quality information in high time. Then building quality into product and enhance understanding and control of manufacturing process. So how to apply PAT, new technologies in pharmaceutical manufacturing? So so in order to apply these PAT technologies in our pharmaceutical manufacturing, so what we do initially, what is to monitor, so that we identify, and what is to measure, that we identify, and how we are identifying those things. We are identifying those things on the basis of the risk assessment. So how to do the risk assessment? So there is also guideline is there, ICH guideline is there. So according to that, we do the risk assessment, and accordingly we decide what to monitor and what to measure. Then after deciding what to measure and how, what to monitor, then we decide how to monitor those parameters and how we can measure those parameters. So to know that monitoring process, like we apply DOE or PAT modeling tool, and then basis to that we got some output, and those output we analyze through some tools, statistical tools, and then we interpret that data, and we can say that whether my process is in control or not, or some improvement is required or not. So what benefits are there? So benefits like enhanced product and process understanding is there by doing this exercise, and then establish critical product attributes and attribute and critical process parameter we can do, and develop design space. We can develop a design space, and this design space we can utilize throughout the product life cycle, and then develop the control strategy for the manufacturing. Okay, so we can develop a control strategy for manufacturing for the future commercial batches manufacturing. Then next is what all are the PAT tools, okay, and how we are applying. What all are the PAT tools and how we are applying in our pharmaceutical manufacturing. So, so I will discuss about the pharmaceutical manufacturing in solid oral dosage form only, because my area of work is in the solid oral dosage form. So here you can see stage-wise it is mentioned. First one is the high CR granulation. Then second one is the dry granulation. Then FDP or FBD drying, and then milling, then blending, then tablet compression and coating. Okay. So then in this high CR granulation, so why we do this high CR granulation? High CR granulation we do in order to densify the powder material so that it will have some adequate flow and adequate compressibility, and we can produce the tablet with a satisfactory quality parameter. So that is the reason why we do the high CR granulation. So in this high CR granulation, our basic objective is to produce the desired characteristic of granules. So it is necessary to measure the granules characteristic, and if you can do this characteristic measurement online, then it is the we can say that it is the best process to have this parameter. Okay. So what we measure through this PAT, we measure this torque, we measure current in the impeller system, how much it is experiencing that difference, and FT4. So these three DFF sensor is also used to measure the endpoint of the granulation. So how this torque is helping? This torque is helping to understand the endpoint of the granulation. Means when we add water or binder solution in the granulation and then we do the granulation, then what happens? Actually, this material get densified, and after a certain time, after a certain time, when we achieve the desired endpoint, at that time we stop the granulation, and then we process for the further activity like drying, milling, and so on. Okay, so the particular endpoint is very critical, and if we target that endpoint, then we can get the desired characteristic of the granules. Okay, and then subsequently we can get the desired characteristic of the tableting property. Okay, then DFF sensor, it is also similar kind of sensor which can help us to detect the granulation endpoint. Then FT4 is a tool where we can measure the powder characteristic. So it is not like during process we can utilize. However, we can use this instrument for our offline testing, and it is very useful test to determine the powder characteristic, and accordingly we can decide the granulation parameters or granulation parameter. Then next one is the dry granulation. So what is dry granulation? Dry granulation is granulation without using water. Okay, and the most commonly used dry granulation is the roller compaction process. So in this roller compaction, actually the tableting property will depend upon the density of the flakes of the roller compaction. Okay, and this density you can measure online. Like for example, so to have this compact, we apply pressure in between two rollers, and then we feed the powder in between these two rollers. Okay. So what happened? So there are many factors. It depends to decide the density of the flakes, and that density of the flakes will finally decide the tableting property. Okay. So this density measurement of this flake is very important, so that we can measure through this gas picnometers. Okay. Then FBD and FBP drying. Okay. So here actually what we do the drying of the wet mass, or sometimes we do the top spray granulations also. Okay. So here actually this drying endpoint actually is to achieve the target LOD. So Okay, so what is our desired parameter to achieve? That is the endpoint of moisture or endpoint of the organic solvent or endpoint of the particle size analysis. Okay, so for example, moisture we want to keep within the 4% of limit. So we can apply some technology inline where we can measure the measure the moisture content in the blend in the granules during the process of drying. Similarly, organic solvent also can be measured. How much solvent is there at the end of drying during the drying process. And third one is the particle size analyzer. So particle size analysis is it is basically we use for the Worcester coating or pellet coating. So there what happen the particle size gradually increases when we do the coating of the pellets. So that gradual increase of coating is very important to observe, and how it is progressing, because it depends upon the many factors. It depends upon the many factors like spray rate, atomization, CFM. Okay, so all these parameters, it is a very depend upon the many multi-factor system. So then particle size measurement and its surface morphology also this icon instrument will give. So this information will be very useful for the process finalization or process scale-up. So this pad tool we use this NIR technology and icon instrument we use for this for the measurement of the moisture, for measurement of the organic solvent. So for moisture and organic solvent we use the NIR technology, and for particle size analysis we use the icon instrument. So then next one is the milling. Milling stage is the milling is one of the common very common unit operation. So this milling process actually we follow for the high CR granulation, and we follow for the dry granulation also. Okay, so main objective of milling is to achieve desired particle size. So this desired particle size online we can measure through the icon instrument. Okay. Then the next stage unit operation is the blending. Okay, so in this blending, actually what we do, we mix different component, different excipients along with the API. Okay, so in case of direct blending, along with the API we mix the excipient, and in case of high CR granulation or dry granulation, we mix certain some excipients with the granules. Okay, so there actually this blend uniformity we measure because that blend uniformity is depend upon some blending parameter like blender occupancy, blender speed, and total blending time. Okay, so online measurement of this blend uniformity through this pad tool will help us to understand whether blend is uniform at the end of our blending or not. If it is not uniform, then we can decide accordingly for the extra blending or something in case of our optimization trial. Okay. So here also we use NIR technology. So this is the technology NIR, but the instrument it is provided by different companies. Okay. So then next one is the tablet compression unit operation. So in the tablet compression unit operation, we can measure the tablet content uniformity, and we can measure the tablet assay also. Okay, through the technology of through NIR technology. Then next one is the coating unit operation. In this coating unit operation, we can measure assay of the tablet, coating thickness of the tablet, and then residual solvent and process humidity. So these parameters we can measure with the different pad tools. Okay. Okay. So what we can measure through the NIR tool? So what we can measure through the NIR tool? Through the NIR tool, we can measure the assay. Okay, and we can measure the residual solvent content. So residual solvent, in case my coating process is the non-aqueous coating, then in that case residual solvent. Otherwise, in case of aqueous solvent, aqueous process, we can measure the moisture content. And by using this OCT instrument, we can measure the tablet thickness. Okay, so coating tablet thickness, coating thickness measurement is important in case of functional coating, where the release is dependent on the thickness of the coating or buildup of the coating. Okay, like many ethyl cellulose coating, many cellulose acetate coating, and all those coatings, actually release is dependent upon the thickness of the coating. Okay, so through this OCT instrument, we can measure the thickness. There are many other technologies also we can measure the thickness through that. Then next one is the I button. I button is the pyro button. So this through this pyro button, we can generate data for the coating process, where we can measure the humidity of the system, and we can measure the real-time temperature of the system. So these all are about the pad tools we can use in the manufacturing of the solid oral dosage unit. Okay. Next one is the digital transformation in the pharmaceutical industry. So digital transformation, I will say that this transformation we can see in the every areas. Okay, and similarly in pharmaceutical industry also we observe many digital transformation in our processes. Okay, so I will go through some examples, and I will explain this transformation process by taking help from our some non-pharmaceutical processes also. Okay, so that will simplify the for our understanding. So first I will go through one by one for the terminologies what we use in the digital process. So first one is the artificial intelligence or machine learning. Okay, so what is artificial intelligence? Artificial intelligence is a branch of computer science that deals with the problem solving by the aid of symbolic programming. Okay, the main objective of this artificial intelligence to identify useful information processing problems and give an abstract account of how to solve them. Artificial intelligence in pharma refers to the use of the automated algorithm to perform tasks which traditionally rely on human intelligence. Okay, so I will give you one example. Artificial intelligence, you can see in a conference room. There is a system, camera system. So that camera system will move towards the speaker, will focus towards the speaker. Okay, if more than 2-3 persons are there in that room, then the camera will focus in that direction, will speak. Okay, so that system is based on the artificial intelligence. Okay, similarly, this machine learning actually machine learning how we are using in the pharma. So I'll tell you. Like for example, we have kind of like inspection process, inspection system, inspection tools we have, like where we do a sorting of the tablet of good and bad tablets. Okay, so there actually we just give that information to machine what is good and what is bad. Okay, and when the tablet it passes through that camera of that machine, and if that camera it will identify that no, this tablet is not fitted as a good tablet in my system programming, so then automatically that tablet will be rejected. So that is the machine learning. So some kind of learning we give to machine, and machine identify those learning, and accordingly it acts on the process. So that is the machine learning. Okay, so I can give you one example. I don't know whether in other cities whether it is implemented or not. So in a traffic control, you can see in a traffic actually this for example in both the side of traffic, it will not equally what I can say, the traffic is not so much equally distributed on all the sides. So this artificial intelligence, what it can do, it can by seeing the traffic of one side, it can decide how much time it should be open for that side and how much time it should be increased for the other side where the traffic is more in the signal. Okay, so for example, in all the sites, it should not be like 15 second, 15 second, 15 second, or 30 second, 30 second, 30 second. If one side the traffic is not heavy, so in that side, that system can automatically recognize, and it can decide that time should be 10 to 15 seconds, and the other side it can be increased from 30 to 40 seconds. Okay, so that is the artificial intelligence application. Okay. So one YouTube is there. I don't know whether time will permit or not. Payal madam, so actually we have started late for our this presentation. So my time is there up to 5 o'clock. So it is not possible to increase it, right?
Yes, you can.
Okay. Okay. So we can extend it. So you can see this video.
Sir, we can't hear you. The video have no sound, sir.
Okay. Just a minute. Let me see.
Share with sound.
Sir, can you share it with sound, the video?
Just a minute. Madam, is it audible?
Yes, your voice is audible, sir, but the video voice, I think it's not.
Sir, if you can do one thing, if you stop share
Okay.
first,
Okay.
then it is still visible, sir. You have to stop share first. Share screen.
Share.
Stop.
Stop sharing.
Sir, you have to stop sharing your screen first.
Okay.
At the top of your screen, you can see a red color button that is stop share.
Okay.
Yes, sir. Now again, you click on the share screen option at the bottom part of your screen.
Okay.
Yes. After that, you have to select the page where the video is there. And at the below of that, at the bottom of, sir, again you have to stop share. Whenever you are clicking, whenever you are selecting the page, there is a check box where you can see it is written share computer sound. You first share.
Okay.
You first click on the share screen option, sir.
Okay.
Yes.
Have to have your video finished.
It is audible. It is audible. It was audible.
This process can be quite long, so streamlining it is strong sound. Hope these things don't later. Now for the recording part, you're gonna need a few things, like some audio equipment, a quiet place, and some props to record your sounds. I recommend you use what you already have, so if you don't have any of these things, don't fret. Using your camera with a shotgun mic, or your phone's audio recording capabilities, will work great in a quiet place. The space where you record your Foley is pretty important. For example, recording your audio in a large garage versus your closet, the acoustics will sound totally different. A great alternative is to use a quiet room and put some kind of sound absorbing material behind you when you're recording, like a blanket or foam. The last thing you're gonna need when recording Foley is your finished film and a way to view it. You'll do this to match the visuals with the audio and help save you some time in the end. The reason you have to have your video finished is because you don't want to do more sound work than you really need to. This process can be quite long, so streamlining it is strongly recommended. Foley can be recorded in two ways. The first way is actually reenacting the actions of the scene, but only recording the sounds. And the second is recording the sounds with other everyday objects. So behind us, this is how we do Foley. We got the TV, my laptop, we got the mic, and the surface we're going to be using to record sound. Now, let's get started. So one of the most basic sounds that Foley artists do are footsteps. I'm going to recreate the footstep sound. I'm going to walk on this tile surface with similar shoes. To recreate the sound, I'll walk in place and match my footsteps with his on screen. One thing I notice about this sound is that he's walking towards the camera. In order to create some depth to it, I'll make my footsteps louder as I get closer to the camera. And one other thing I have to mention is that the surface in which you create a sound really matters, not only for footsteps but pretty much all Foley. I suggest to always use materials that will make sounds that suit the visuals. Now for something more complex than footsteps, I have this action scene. I'm going to make some punching sounds from scratch. So there's a lot going on in this scene, and I won't be able to record it all in one take like I did with the footsteps. So what I'm going to do is record the sounds separately and then mix them together. A lot of what I'm going to use are food products. The reason is I need some sounds to sound like human flesh or bones breaking. And food products are really good at making sounds sound like human flesh. So instead of eating dinner tonight, I'm going to use this chicken and celery to make some action sounds. So what I'm going to do is I'm going to take this chicken, I'm going to beat it a bunch of different ways to get a variety of sounds and then match them up with the clips later. Let's do it. So for this last hit, I really want to make it seem like it did a toll on the guy. So when his neck turns, I'm going to use the celery to make it sound like it's breaking. So the chicken sounded pretty good, but it's only really going to work.
So what actually it is telling in this video. So what we can understand from this video actually, if you can see that camera is moving towards the sound.
Have you ever noticed the sound?
Camera. I don't know whether you can hear me or not. Payal.
You can go to the next slide. I think it's.
Have you ever noticed?
Go to next.
Sir, you can go to the next slide.
Okay.
And you have to share the screen. Share screen. Yes.
The sound design.
So then next one is the blockchain. So this is about your artificial intelligence and machine learning, where actually camera is focusing towards the sound. Okay, and then next one is the blockchain. So blockchain here actually blockchain we are using for this our for the supply chain purpose of our these medicines, where we can trace the trace our medicine supplying. Okay, and we can block the flow of counterfeit medicines and substandard drugs. Okay, which makes their way through the pharmaceutical supply chains, killing thousands of patients every year. So, so there are many fraud companies who can sell these medicines in the market. So, in order to block these activities, we can use this blockchain process. So how we can block this and how we can implement this. So this is the process of serialization. So this is the process of serialization. So that we will explain in subsequent slides. This is also one video. I don't know whether time will permit or not.
What if?
So I am skipping this video. Then next one is the digital training or digitalization in the documentation activity. So what is that digital training? So this is like previously actually we do that training in the classroom training. Okay, so nowadays this many companies, they are transforming this training into the digital training process. And the digitization in the documentation activity. What is this digitization in the documentation activity? So in pharmaceuticals, there are many documentation where many CFPs, they review this document. But nowadays this documentation we do online, and the signing and review of this document is happening to the system and online. Okay, so there are many companies who develop many of such kind of software. So where we can upload this document for the collaborative review and approval of the document. And second thing is that there are many documents like executions documents are also there. So those documents also we do in such a way like online recording of the process parameter and all those things can happen. Okay, so this is the digitization of the documentation activity. Then what is the cloud technology? So here actually we use this cloud technology to store our data, okay, and use those data for our data analytics. So and the use of various cloud application has also improved in CGMP compliance. And when regulator come, then we can store this data directly from the clouds, and then which meet the standard of the regulatory guideline and quality guidelines. Okay, so this technology is a boon for industry as it eases the flow of information and reduces the chances of error. Another aspect of cloud technology is the use of the electronic signature. This legally binding e-signatures streamline data related processes. Okay. Then next one is the big data and analytics. So through the digital process, we can analyze the data and we can represent this data in different form, and which will help in many areas of the pharmaceutical process like investigations, trend data analysis, and all those things. So there also we are using this digital process. Now next, these are some digital processes where we are using our this digital process for the specific areas of the drug development or drug research or the pharmaceutical manufacturing. So first one is the smart drug development. So here actually in smart drug development, one process is that in vivo AI drug discovery system. So what is in vivo AI drug discovery system? So here actually artificial intelligence process actually we use to discover the drug molecule. From the data bank of the drug discovery, this artificial intelligence they can identify, and whatever the required action we require. So accordingly, this drug molecule can be developed through this AI system. Then Pangea data, patient cohort identification. So this is a system actually. They use the data bank of the patient, where if we want to use some specific kind of patient for the clinical trial or clinical trial or bio study, so then we can pull those patients and we can do the trial. So that is the Pangea data process, patient cohort identification system. Then smart API development. Smart API development is the smart API development. Here actually we utilize Sequoia technologies, that is a continuous manufacturing optimization for the API development or API manufacturing. AI reaction prediction. AI reaction prediction is artificial intelligence reaction predictions. So in this AI reaction predictions, what happened like in the data bank there we can store many reactions and during the API development we can choose the particular reaction which is useful to produce effectively the API with our desired characteristics like low impurity, high yield, and all those things. Then next one is the smart formulation development. Smart formulation development, that is the POMICEL in silico modeling. Okay, so POMICEL in silico modeling. So this modeling actually we can use for the development of the formulation development. So this will predict how much quantity of the disintegrant or binder we can use to have our desired drug release profile. Okay, so this is the POMICEL in silico modeling. Then smart analytical program. Smart analytical program is the analytical program where we can use our smart thinking and the AI system. So in this smart analytical program, actually we can use for the shelf-life prediction by using the small data, and we can use this smart analytical program for the drug product characterization for reverse engineering. Like for example, we have one marketed innovator product, and we want to know how much concentration of Agdisol is there in that formulation, or how much concentration of binder is there in that formulation. So we can use this smart analytical program to characterize, to identify the concentration of the disintegrant or the binder or the rate controlling polymer in the formulation. Okay, this is a smart analytical program. Then smart clinical trial program. So this is a smart clinical trial program. How we can use this? This is an in vitro release prediction process based on the CMA and CPP of the drug product. Okay. So based on the material characteristic and the material property, like for example, we have many formulations where we use different binders, different lubricant and glidant. So in vitro release can be predicted by using those data bank, and can be mentioned that how much quantity or how much concentration of the that functional excipient can be used to have the desired in vitro release profile. Then in vivo prediction based on the PK data of the molecules. So this also we can use for the clinical trial program. Okay. Then precision medicine. What is the precision medicine? This precision medicine means personalized medicine. So there are some medicines which we can apply for the particular person or particular patient. Okay. So there are different tools available in the market. Or that is an exact cure, that is a drug exposure model study we can do through this software. Then TEPHRA is the individual cancer vaccines deciding tool. Okay. Then next one is the additive manufacturing. So additive, what is additive manufacturing? Additive manufacturing is the 3D printing process. Okay, where customized way we can do the 3D printing, and we can use that 3D printed peel for the patient. So Fabex is the available program in the market. By using that, we can do that 3D printing and printed peel to use for patient. Then Frontier Bio is a tissue bio printer. Okay, and then so this is about the additive printing. And then next one is the blockchain. So blockchain, as I said, that it is useful to trace the distributed medicine, and it will help us to avoid the distribution of the any adulterated medicines or duplicate medicine in the market. Okay, so these are two tools. Like one is the Pharma Trace, that is a smart contracts tool, and then VeraTrack. VeraTrack is the pharma supply chain tool. So what they do actually, they do the tracing of the medicine, tracing of the container, how it is shipped, and how much exposure of the temperature, how much was the exposure of the light, and all those things. It can be traced out. So once it reaches to the destination, so then after that we can extract all the data, and by extracting all the data we can see whether this transportation was happen as per the compliance or not, whether there is any excursion of the temperature or not, and all those things we can be this information can be extracted through this using this software Pharmatrace and Virattrace. Okay, so this is called the blockchain. The next one is the digital therapeutics. So digital therapeutics is there are two systems now it is in the market. That is the Cognivive and DopaVision. So DopaVision, Cognivive is the neuro rehabilitation system. So like for example, this utilizing using this system, like if some patient is in depression or something, so then using this digital process, we can apply to the patient, and the patient can feel that that he is in resort or some areas where he can feel better, and he can come out from the stage of depression. Okay, so that is the Cognivive software we can use for the digital therapeutics. Then next one is the DopaVision. So DopaVision also we use for the eye treatment of the, I think it is for the for the myopia, I think so. Okay, so these all are the two. So these all are the digital tools we use in the pharmaceutical system. Okay, so in my area actually we are not using all those things. Okay, so as it is many expanded like digital therapeutics, it is I am not using. Okay, so blockchain for every pharma distribution they are following this thing. So nowadays this serialization is compulsory from the regulatory agency. So so all companies they are following this blockchain system. Okay, so Payal, actually I think it is not possible to explain more about. Actually I have prepared this in detail slide for this explaining all these tools, but probably it is not possible to explain now because time is already 5:07 over.
Sir, I think you can go a little brief with those slides and you can continue because we still have almost like 15-20 minutes where you can continue.
Okay, so you mean to say I can continue, right?
Yes, you can.
Okay.
At least
Okay. So this DopaVision I skip. So I will tell you, like DopaVision treatment is a German startup company. DopaVision is making a smartphone based digital therapeutic for myopia. Okay, and the startup solution aims to slow down the progression of the myopia in the young population, especially in the children. The solution achieves the activation of the dopamine, a neurotransmitter that plays an important role in eye growth regulation. Okay, so DopaVision is currently undertaking preclinical trial of digital therapeutics. So it is not in the market, but it is in the preclinical trial. And actually this concept is very new concept. So many of the things you may not find in the market, and this all are in the development stage. Okay. So Cognivive already I explained. This is the neurohabilitation. So Cognivive, a US-based startup company. This company offers the evidence-based digital therapeutics for the treatment of neurocognitive and neuromotor impairment. Okay, its neuro rehabilitation treatment uses the medical devices to help patient recover from a stroke. Okay, and cognitive VR, the startup's VR headset, further immerses individuals in a virtual resort world. Consequently, the brain develops new brain circuits, helping in the recovery of the brain-body control. Okay. Then digital therapeutics deliver evidence-based therapeutics intervention using the software to prevent, manage, or treat physical, mental, and behavioral conditions. So these non-pharmacological technology-driven solutions are either standalone or used along with the medication devices. So these two are the example of the digital therapeutics. Okay. Then additive manufacturing. So additive manufacturing is the 3D printers. So where actually multiple layers of printing we do for our therapeutics purpose. Okay, this is the main objective. So what is this? So a lot of research is underway for making advancement in the 3D printer that print tissue or cells. 3D printing of human tissues has great applications in drug development, organ engineering, and regenerative medicine. So this allows the development of age or physiology dependent medical formulations as well as precision pills. Okay. So bioprinter also help in pushing the innovation in bioinks, tissue engineering, and microfluidics. Okay. So two tools are available now. That is the FabRx and the Frontier Bio. Okay. So FabRx is the printed pill. It is a FabRx produces the printed pill. It is a British startup company. So that manufacture M3 dye maker, a 3D printer for making personalized pills. So M3 dye maker uses proprietary technology for direct powder extrusion. The single-step printing process uses a single screw extruder for the extrusion of the powder material. It manufactures pill with properties such as the sustained or the delayed dose and multi-drug combination pills, polypills. The printer also enables the small batch productions for clinical trials and precise personalized dosage forms for individual. Okay, so this is the FabX. Okay, FabX printed pill, and then Frontier Bio is the tissue bioprinter. This is also a US-based startup company. This also produces a 3D bioprinting for making the human testing. Okay. Then Exact Cure is a drug exposure model. Okay. So Exact Cure, a French startup, offers a software solution for simulating the effect of drugs in a patient's body based on personal characteristics. Okay. So a specific drug, when it is required for a patient based on his personal characteristics, then we can use this Exact Cure, this tool, software solution. The startup makes use of the population pharmacokinetics as well as scientific literature data for real-time prediction of the efficacy and drug interaction on each individual. So as I said, that data bank, using the data bank of the previous patient and all details, so they can predict the treatment for the particular patient required for that disease. Okay, so Exact Cure is developing drug-specific exposure model for drug under investigation for the treatment of COVID-19. Similarly, Tephra is an individualized cancer vaccine. So it is a Swiss startup. Tephra offers the platform technologies for the identification of the T-cell antigen. Okay. So this is also a precision type of precision medicine we can use. Okay. Then Pomicell in silico modeling. So this is we use for the smart formulation development. Okay. So this is a startup, Israeli startup. Pomicell offers software tools for big data analytics in the pharmaceutical research and development. The startup utilizes the machine learning techniques to analyze and aid in development of in silico models. The startup further builds a customized drug development roadmap by augmenting the available data analysis and insight through the matching and in silico model. So as I said, that if you can build a data bank where we can say that this much concentration of a substance, this much concentration of a polymer, or this much concentration of a disintegrant can give this type of release, and we can make a huge data bank for that. So then we can use some modeling. So where we can decide the required concentration for those particular material to have our desired release characteristic. So this is the Commercial in silico modeling. This is the software tool where we can use for the formulation development. Then Sequoia Technologies, it is a continuous manufacturing optimization. So it is a Belgium startup company. So here actually this is utilized for the API manufacturing. Okay, so this offers the novel technologies for production process by tailoring the continuous manufacturing process equipment down to the ideal scale. So microstructured elements such as the microfluidic droplets generator are used for optimizing manufacturing processes. The startup solution find use in the intensified chemical synthesis, crystallization, pre-vaporization, and microencapsulation. So this is one of the technology where we can use the API manufacturing. Okay. Then next one is the in vivo AI drug discovery. So in vivo AI drug discovery is a Canadian startup company. They developed this in vivo AI tool. Okay. So which developed the novel algorithm for the drug discovery. Okay. Okay. So the startup uses different machine learning approaches, such as the few sort learning, reinforcement learning, active learning, and representation learning to aid the drug discovery process. Okay, so this is the process by which we can discover the new molecule, which will have some less or least toxicity, and we can use for the further of the clinical trial. The deep learning solution uses small and noisy data set to predict and optimize the potential drug candidate, further eliminating the need for the large data set. Okay, so usually what happened during the drug discovery, we manufacture wide spectrum of molecule, and then screening and tailoring of those molecules take much time. So we can reduce those time by using this software. The next one is the Panagia data patient cohort identification. I already explained it. So this is a data bank of the patient, so which we can utilize to have our particular clinical trial. So it is a British startup that uses the unsupervised AI algorithm to identify the patient cohort for the drug discovery, clinical trial, and real-world evidences. Okay, real-world evidence study. So the machine learning-based software scans through the electronic health record and under unstructured doctor's note to find the right patient based on phenotype. The startup also develops a library of AI models for the different disease areas. Okay. So this is all about the details of the digital tools what we are using, or digital process what we are using in our pharmaceutical industry, and this is the recent trend. Okay. So now the question answer session. So thank you all for your presence, for listening me. Okay.
Thank you, sir, so much for your nice presentation. So from the session, I have two questions to ask you. So as you mentioned, artificial intelligence and machine learning in this process, can you a little bit elaborate us how we can execute that in this different kind of methods, applications of that, where you are using exactly where you are using this AI and the machine learning techniques? Can you a little bit elaborate on this part?
Machine learning and artificial intelligence in our pharmaceutical industry, you are asking?
Yes, because I think you showed a slide where with this conception you are using this artificial intelligence and the machine learning in this method technology.
Okay, so machine learning, as I said that we commonly use in our inspection system of our tablet. Okay, inspection of the tablet, inspection of our tablet or capsule. So where we can screen our good or bad tablet. Okay, like for example, I will say like there is some defective tablet. Okay, like printing not done properly, or there is some film cracks in the film, or some kind of erosion in the edges. So those kind of tablet can be rejected if we pass through a camera system, and if we can set some logic in the camera that this is a good tablet and this is a bad tablet. So we have to provide some input to the camera by providing some images of good tablet and bad tablet, so that it can identify what is when the set of tablet will pass through that camera, so that it can identify what is which one is the good and which one is the bad tablet. Okay, accordingly it will sort out. Accordingly it will reject.
So you are already setting the parameters in the form of picture mode where it can detect through the camera.
Yes. Yes.
Okay. Okay.
Yes.
Thank you, sir. So I have the chat box open for the participants who can please write down your questions for this session if you have, sir, so I can mediate it. Please write down your questions on the chat box so that we can answer for the session.
So, Mr. Lahari, am I audible?
Yeah.
Yeah, actually, this PAT techniques used for reduction of cost in production department as well as in formulation development section, right?
Yes.
To improve that product efficiency output of the manufacturing. And QBD is also the concept of improving the efficiency of production.
Yes.
QBD. So what basic difference between PAT and QBD? Because QBD is also used to define that attributes like critical quality attributes, material attributes, product attributes. So by optimizing those quality attributes, material attributes, we can justify the good quality of product and also reduce the cost and output of the production can be hastened. So what basic difference between PAT and this QBD concept? And this PAT, whether it is implemented or is a guideline of FDA or ICH?
Which one? PAT.
PAT, yes. Is
Yeah,
it
so
coming
guideline
as
is there. Guideline is there. Okay. How even recent one more guideline came in the FDA side. So that is how we can use for the NR tool for our this blend uniformity or residual solvent analysis. So those guidelines are there in that FDA side. Okay. And coming to your first point, what is the basic difference between the QBD and the PAT? So
yes.
PAT actually it is a it is a process analytical technology. So which we can apply to have our quality.
When we are preparing some granule by following dry granulation or wet granulation, so we have to measure the moisture content and that flow. So
Yes.
that we also can perform by simultaneously by performing some in-process testing. Okay, in the production.
Okay.
So what basic difference that PAT adapting PAT and this QBD?
Okay. So
Is there any basic advantage of?
So yes, you said rightly that we check the LOD of the granules after drying. Okay, in our IR moisture balance, but that is the offline checking.
Yes.
Okay, and what we do, we stop the process and we collect the sample and then we check the LOD, and if the LOD is more than the limit, and then we dry it further. But by utilizing the PAT process, what we can do, we can do the checking of LOD or water content online. Okay. And there will be no interruption in the process in between the drying. And once the target LOD or water content will achieve, and then we can stop the process. That is the concept. And similarly, like I said that residual solvent also can be measured or estimated during the process of the drying. Okay, so you know that the residual solvent actually we test in QC with the GC method, and which is a very time-consuming method. And so where what we do, like we check the LOD. So LOD is not the direct measurement of this residual solvent, and we send sample for the residual solvent testing in the QC. So there is a stoppage in the process, and when the residual solvent will come, and if the solvent is out of limit, so then again we have to dry that material. So that process is not acceptable, redrying of the material. So instead of that, if you can use the pad tool, and if you can feed the pad tool in the flange of our this FBD, and then we can measure the residual solvent online, and when it will reach our desired limit, so then we can stop the drying. So that is the main advantage.
Okay, means PAT is totally an online process.
Yes, it is. So that's what the expectation from the regulatory agency, that you do this testing online rather than doing it in the offline and stoppages in the process. So that will give the more robust and but reproducible process. That is the expectation.
Thank you for your nice explanation. Thank you.
Thank you, Tawan sir, for your questions. So we have another question from one of our faculty, Shormisha Pal. So her question is, why can't we see any AI-based startup from India, and what would be the possible reasons for this? And please share your view on this. That's what she suggested. She asked for from the speaker.
Okay, so this is very emerging area, and so I already mentioned that many companies actually many of these tools are already in the clinical trial stage or something. So this is a concept actually developed in last 2 years, mainly 2-3 years. So in India, I don't think any companies, they have started thinking on this way, or they are doing this digital process.
So another last question from this session is from Snehanshu Vishas, who is also a participant for the session. His question is multi-layer printing and 3D printing also being used by the manufacturing unit to trace the genuinity of their products in the market?
3D printing actually is not to trace the genuinity of the product. So to trace the genuinity, so to trace the genuinity of the product, we use the blockchain process. Okay. So that ensures the correct supply chain of the product, and we can understand whether this product is product available in the market is genuine or not.
Okay, sir. Thank you for all of your participations. Here we have shared the link for the feedback form, and it will be open for the next 1 hour, where you can feed in your answers, and you need to have minimum of 80% from the answers to qualify for the FDP certificate. And thanking you once again, thanking you, sir, for your wonderful session. And I hope it was beneficial to all of us attending it. And thanks from all of us for your valuable time. And we like to end up this FDP session for success. And here we like to show the certificate to our speaker. Thank you, sir. This is your certificate of appreciation. Sure. So, and we are going to send this to you, sir, also.
Okay, thank you, madam. Thank you for your this appreciation, and thank you all our GIS team Okay, for giving me this opportunity to share my experience. Okay. Thank you all.
We are ending the session tomorrow. And tomorrow 3 p.m. tomorrow as well, we will be meeting for the FDP session. Thanks for all your participation.
এখন ওইটা বন্ধ করো।